So in Module 8b, we're going to talk about a couple of connections between the chi-square goodness of fit test, which we talked about in 7a, and the binomial distribution. And this is a constant source of confusion because there are two very different connections between these two tests, or between these two concepts. First of all, we can use the chi-square goodness of fit test instead of doing a binomial test, right? And so there's going to be a way to approximate the results of a binomial test with much less calculation. It's not going to be exact. It's going to be only an approximation of the p-value, but it can be useful. And so I'm going to talk about that first. And then the next part of this module, I'm going to talk about cases where we want to test a null hypothesis where the null hypothesis calculations come from the null. So it's really common that we have a null hypothesis that there are multiple trials and they all have the same probability of success. So our data is in some kind of count. So we have some sample size n sub s where the data is the number of successes on some number of trials, which can be different than n sub s. And then we get a binomial distribution that gives us the null expectations for the chi-square test. So in one case, we're using the binomial to get null expectations, and in another case, we're using the chi-square to substitute instead of doing a binomial test just because it's easier. All right, so let's just walk through these in turn. In the in-class exercise 7b, we had a situation thinking about ESP and detecting it with these 5 different cards and whether someone could concentrate on a card and another person could receive their signal telepathically and guess what card they were looking at. And there were 5 different cards, so the probability of success was 0.2 if you were just randomly guessing. And we had some question on that in-class exercise of like assume another extremely hypothetical individual tried to guess ESP cards and got it, they tried 1,000 times and they were correct 350 times. And in class, we didn't do a hypothesis test to see if this was better than random. We did a confidence interval because it was just too tedious to sum up all the tails of the binomial distribution that would contribute to the p-value. So we didn't get a p-value. We just had to make an argument of like, well, the null hypothesized value of 0.2 was outside of our confidence interval, so we'd probably reject. But if we think about that, okay, the null hypothesis is that the probability of getting the card correctly is 0.2. The alternative is just that it does not equal 0.2. We have a sample size of 1,000. We had 350 successes. We can sort of rearrange those in terms of the expectations and the observed number of successes. So here's what I just showed on the previous slide. We can think of the number of successes as observation 1, 350 of those, and observation 2, observed quantity 2 is n minus observation 1, so that's 1,000 minus 350, so that gives us 650. And we have in our null hypothesis a way to calculate expectations for both of those. So E sub 1 is n times p, that's 1,000 times 0.2, or 200. That's how many we expect to get right by chance. And the expectation for 2, this should be a 2 here in this subscript. Sorry about that. I'm just going to put that in like that, is the sample size times the probability of failure. That was 80%, 0.8. So 1,000 times 0.8 is 800. So it seems like, okay, this naturally was a binomial problem, but we could also view it as a case where we have an expected number of successes and observed number of successes. So why can't we do this as a chi-square? And we can. We can set up a chi-square table just like we did in the previous module with the observations and the expectations under the null hypothesis. Calculated just the numbers that I showed on the previous slide. I slightly changed the indexing here from 0 and 1, but regardless, it's the same information I showed. So how would we calculate the chi-square test statistic? We would subtract those two. The difference between these two is 650 minus 800 is negative 150. We would square that and divide by 800. And over here, 150 minus 200 is 150 squared divided by 200. Remember, I talked about before you do the squaring, everything should add up to 0. So when you're using a chi-square to perform a binomial test, it's nice because there's only 2 categories, so the numerator should be the same magnitude, but just opposite signs, right? So if we do those calculations, we end up with 28 and 112, right? They're the same numerator, but the denominators are very different. So we have a much larger chi-square deviation for this category of successes. 112 is our chi-square term there. So if we add those up, remember, we get the chi-square test statistic by adding up all of these observed minus expected squared over expected. We get 140. Okay, remember, if we want to decide whether that's a significant result, we have to compare that test statistic value to the null distribution. And in order to know the null distribution, we have to know exactly what form of chi-square we're talking about. So we need to know the degrees of freedom. So the degrees of freedom is number of categories, k, minus the number of parameters we estimated from our testing data to get the expectations for our categories. We didn't do any of that in this case. The null hypothesis specified what the probability of success was. We didn't estimate that from our data. We came in knowing it was 0.2. So that's going to be 0. There were 2 categories here. We subtract off 1, and we get 1 degree of freedom. And remember, this sort of makes sense because the data can surprise us here. We were expecting 800, we got 650. That could be surprising. But once we know we have 650 in our sample size of 1,000, we know that the other result has got to be 350. So there's only kind of one dimension in which the data can surprise us, so the degrees of freedom are just 1. Okay? And so we look at the chi-square table. We only have to look at the first row of that table. Remember, the table is organized by degrees of freedom. And so we would say, okay, we got 140 for our test statistic value. We would go and look at the columns that we use for hypothesis testing. At the 0.05 level, the critical value is 3.84. So I wrote that down here. Our value is larger than that, so we know we would reject at the 0.05 level. Maybe we can make an even stronger rejection. So at the 0.025 level, the critical value is 5.02. Our value is still larger than that. So we continue moving rightward on the table. 6.63 is the next critical value, then 7.88, and then 10.83. And that's as far as our table goes. Our chi-square test statistic calculated on our real data is larger than 10.83. So that means that our p-value is smaller than this alpha value right here. So it's this weird kind of inversion. If our test statistic value is more extreme, more in the tail of the distribution, then we know the p-value is going to be even smaller than the alpha value associated with that critical value. Okay, so if we think about what the chi-square distribution looks like, this is a chi-square for degree of freedom 1. These are those critical values that I pulled out of the table before. There's the 5%, the 2.5%, the 1%, etc. And our value is way over here to the right of all of these. And so clearly the probability of getting a value of 140 or greater is really, really tiny. So we would summarize that, say, in 1,000 trials and imaginary receiver. This really wasn't real data. This was just made up data. Correctly guessed 350 cards. This is significantly more. And since we said significance, statistical significance, we want to quantify that in terms of the test statistic use, chi-squared equals 140, degrees of freedom 1, and the p-value. That's how we're judging significance. All right, so that was significantly more than expected value, and expectation was 20% correct if the participant were guessing at random. So that might be a reasonable conclusion for that. It gives us the sample size, it gives us our summary statistics, our test statistics, and our p-value. So we're being super clear while also being pretty concise. Okay, so hopefully that makes sense to folks. If I go into R, I can actually calculate the p-value more exactly using the chi-square approximation. It was 2 times 10 to the negative 32. I can actually in R also calculate the p-value using the binomial distribution, and there it's 8 times 10 to the negative 29 for these data. So we can see that these are not the same number. That's because the chi-square is really just an approximation of the binomial, and it's off, almost by, 4,000 here. But it gets us in the ballpark in terms of making our decision whether we accept or reject. These are both tiny p-values, so we would firmly reject the null hypothesis in either case. So we're using the chi-square in this case because it requires less computation. It's appropriate because this is a random sample and our data is counts of categorical variable with large expectations, large expected numbers, right? We didn't have any expected numbers less than 1 or 5, so we can use the chi-square in this context. But remember, the association between the chi-square test statistic and the chi-square distribution is really only approximate until you get to enormous sample sizes. So we would prefer to use the binomial distribution in these contexts because the binomial is exact. But if we run out of time, we can't do it in terms of the computation. The chi-square gives a pretty good approximation if we're checking all these boxes. And of course, if we don't have a random sample, we can't really do the binomial either. So it's a good approximation to have up your sleeve. And this is a really common theme that we see in a lot of statistics, that there's more than one way to analyze the data. There's often a preferred way, but another way may be more applicable in terms of computational or other considerations. And so it's useful to realize some connections between different types of tests and say, this data could be analyzed in more than one way, and whichever one's most convenient for me, I can validly use. Okay, so Hope that makes sense to folks about how we can use the chi-square to approximate the p-value for a binomial. Okay, a second but really different connection between the binomial distribution and the chi-square is often we use the binomial to get expectations for the null hypotheses. So once again, digging back into a problem we've seen before, a question taken from an exam in this class, in a tetraploid plant, each individual has 4 copies of each autosomal gene. An autosomal locus for a color has 2 alleles, big A and little a. 60% of the alleles are big A, that's the dominant allele, which leads to yellow flowers. Plants having only the little a allele, so all 4 copies are little a, are going to be white. And this was the question on the test. If mating and survival are random with respect to the trait and locus, what proportion of these tetraploid plants will be yellow? Okay, so we've seen that question before. Hopefully you remember it from exam 1. Let's take a slight tweak on this. So this is the same background information as I showed on the previous slide. Let's say the question's been slightly changed. A researcher wants to know if there's evidence for non-random mating or survival at the locus. So she genotypes 38 individuals in the population. The null hypothesis is the genotypes occur in frequencies according to the 4 independent draws from the gene pool. So there's just 4 independent draws. That would be what we would get if there was no kind of preference for one particular genotype over another. We're just treating the gene pool as the source of all of our gene copies in an individual. And then the alternative is just the genotypes differ from the frequencies of those 4 independent draws. Okay, so that's a very precise null, which is what we want to see. And the alternative is everything else, which is also what we want to see. And we can say, okay, well, this looks like a chi-square type problem because we've got 5 different types of genotypes. Here I've got an index. This is her data. She observes 2 individuals have 4 copies of little a. 8 individuals just have big A, and then 3 copies of little a. And remember, the way we normally do genotypes in genetics is we don't worry about the order of these because there's no real order to the chromosomes. We just say, you got 1 big A and 3 little a, and we often sort of alphabetize the lowercase after the big ones, but that's meaningless. But the point is that these 8 individuals had 1 copy of the big A and 3 copies of the little a. And then there were 11 individuals that had 2 of each, 10 individuals that had 3 big A's, and 7 individuals that were homozygous for the big A allele. They had 4 copies of the big A allele. Okay, so that's the observed data. If we were going to do a chi-square, we need to get the expected data. So if we look back at our null hypothesis, it says the genotypes occur in frequencies according to 4 independent draws from the gene pool with this probability of a big A allele. Well, that doesn't give us exactly these numbers right here. It just says something about how these numbers should be generated. And so genotypes occurring in frequencies according to 4 independent draws with the same probability of success on each draw, well, that sounds like a binomial type problem. So it's just a situation where the expected numbers for the different categories come from the binomial distribution. So we're going to use a chi-square test to evaluate whether our data fit the expectations, but the expectations themselves are coming from a binomial. So the expectation, for instance, for category 1, that's the expected under the null of how many individuals that would be little a, little a, little a, little a. So 4 little a alleles. That's our sample size times the probability of getting an individual in that category. So if we just expand this p sub 1 to what it is the probability of, it's our sample size times the probability that the genotype equals the 4 little a alleles. Remember, n is the number of individuals that she's genotyped, so n times p 1 is going to give us the expected number there. But so n is the sample size for a study. That was 38. But there's also 4 samplings from the gene pool here, so we've got sort of another sample size kind of going on, and each of the events now refers to a sample size of 4. So that's the draws from the gene pool. It's not the number of individuals she has sampled. So it's useful to start thinking about, well, maybe we want to subscript the sample size. So n sub s is her sample size, that's 38. N sub g is the number of genes in an individual, that's 4. And this is going to be the sample size for the chi-square, n sub s, and n sub g is going to be the sample size for the binomial expectation. So I realize this gets a little hairy, but it is useful to have nice clear notation here. So let's just continue working through the expectation for this first category. This is the same as what was on the previous slide, actually all the way down to here. What is the probability that the genotype would be 4 little a alleles? That's the probability that we have 0 successes, where a success is drawing a big A allele, and the probability of a success is 0.6. So what's the probability of 0 successes, given that we have 4 draws, and the probability of success on each draw is 0.6? So from the binomial, that's n choose k, or in this case, n sub g choose 0. That's our k value that we're interested in. So the probability of success raised to the 0 power, probability of failure raised to the n sub g minus 0. Right? So if I put in numbers here, it's 4 choose 0. That ends up being 1. 0.6 to the 0 power, that also ends up being 1. And then 0.4 to the 4th power is this number here. So that's the probability that if this population were described by random draws, 4 random draws from the gene pool, that's the probability you'd see 4 little a alleles, homozygous for the little a in a tetraploid individual. Okay, that's not the expectation for the how many that this person would see when she did the researcher would see when she collected 38 individuals. That her expectation is her sample size times that probability. So 38 times the probability of success on each trial. So that's 0.9728. So that's just the expectation for the first category. So if we go back to our chi-square table, all of that work is doing this binomial probability here and multiplying it by the sample size, her sample size of 38, that gave us 0.97. In order to fill out the expectations, we'd need to do that for the other 4 categories, right? So using the binomial here, the only thing that's changing, they all have the n sub s, the sample size for the chi-square test put out in front, but what's changing is the k, or the x value, is going from 0 up to 4 for how many big A alleles were found. So like here's a genotype with 1 big A allele, so it's n sub g choose 1, 1 success, 3 failures, 2 successes, 2 failures for the next one, 3 success, 1 failure for the third one, etc. So I went ahead and just plugged through this math. You can check yourselves on any of these, but it's just a binomial formula multiplied by the how many individuals were drawn from this population to give an expected number that would be in each of these categories. So we're using the binomial here, but we're not testing the binomial. We're using that as an expectation. We're going to do a chi-square test, as that's where we're going to get our p-value, is what I mean by that. Okay, so those are the expected numbers. Looks like we're ready to do our chi-square. I just threw away the formulas and just showed the equations here. If we add them all up, we get 30, excuse me, not the equations, but the numbers here. If we add them all up, we get 38, which is our sample size, so that's good. Seems like we're ready to calculate the chi-square terms and sum them up to get the chi-square test statistic, right? Well, not so fast. Remember, one of the issues is with the chi-squares when we have small sample sizes. So this first category has an expected number less than 1. It's not much less than 1. We saw 2, but we would expect less than 1. And remember, this is a slide from the 7a module. When the null hypothesis is true, the chi-squared statistic approximately follows the chi-square distribution with the appropriate degrees of freedom for large n. But if any category has an expected number less than 1, or 20% of the categories have expected numbers less than 5, then we need to combine categories until neither of those conditions are true. So we've got a situation where one of our categories is less than 1 here. So we need to do some grouping, some clumping of categories, coarsen our data a little bit. And so what we typically do is bend together 2 adjacent categories. So instead of 1 and 2, we're just going to merge these 2. And that's going to change my indexing here. So now we just have a total of 4. And I haven't done anything other than just remove the horizontal line here to say that these 2 are going to get added together. So when we add them together, we're going to add together the expected, but also the observed data, right? So we saw 2 individuals in this genotype and 8 individuals in this genotype, so 10 individuals overall that were one of those 2 genotypes, but the expectation is the sum of 0.97 and 5.84. So we would summarize that as like, okay, this first category now is like you've got at least 3 little a alleles. The 4th allele could be big A or little a, 4th gene copy. So the total observed was 10. You expected 6.81. And now we look at our table, and we've got one value less than 5. That is technically over 20%, but I'm just going to let that slide at this point. I suppose I should have bent some more But don't do this on an exam, but I'm going to let myself slide. But apart from that, we got rid of the one that was the extreme outlier that had a less than 1. Okay, so probably should do a little more bending in this case, but we'll just show how to go through this, ignoring that detail at this point. So if we do that, we calculate the chi-square test statistic, how you expect, 10 minus 6.8, and we square that, divide by the expectation. We do that for all of these. Remember that if we do the subtraction, we can check ourselves that all of these numbers should add up to 0 because those are deviations. Before we square them, they add up to 0. So they do in this case. We go ahead and plug through the calculations, and we get these numbers here, where the first bin, which was originally 2 bins in our data, is the largest deviation, the largest chi-squared term, but overall the sum is quite small, 3.46. Okay, so that's our chi-square test statistic, accepting the fact that we probably shouldn't have done it because of that low expectation here. Let's not worry about that right now, but hopefully the rest of this makes sense. How do we calculate the degrees of freedom? Well, originally we had 5 data types, but we're doing the chi-squared test on these data in front of us, so we need to treat that as the number of categories as being 4, not 5, because when the data that we actually applied the test to only had 4 categories. The number of parameters estimated to get the expectations, well, that was 0, because remember those binomial calculations, we knew going in the probability of a big A allele being present was 60%, and we knew that they were tetraploid 4. So we can get all those probability calculations from the background assumptions, not from the data that we observed. And so we didn't estimate any parameters in order to come up with our expectations. We just had to do a lot of binomial calculations. Okay, and then we always subtract off 1 there, so it's 4 minus 1 is 3. So 3 degrees of freedom. And our chi-square test statistic is 3.46. And when you see chi-squareds terms that are less than 1, that usually indicates a really, this one's slightly over 1, but the others are less than 1, really minor deviations. So we end up with a pretty small chi-square test statistic. Are we going to reject the null hypothesis? We don't generally reject when we have small test statistics, but the way we make that rejection decision is by looking at the table or looking at the distribution. So we're here in the third row of the table, degrees of freedom 3. The chi-squared critical value at the 0.05 level is 7.81. So our test statistic, 3.46, is not even as extreme as that. So because our test statistic is smaller than the critical value, our p-value is larger than the alpha that's associated with that critical value, right? So p is greater than 0.05. Using R, I could calculate the p more or less exactly, and it's 0.33, so it's quite a bit greater than 0.05 for this example. So here's the chi-square with some critical values for the 5%, 2.5%, 1%, and 0.05% shaded in different colors. Here's that same chi-square distribution, but this is our test statistic of, what was it? It was 3.46, I believe. So this is 3.46 shaded in. You can see how much probability is down here. You can see that it's large. You wouldn't be able to eyeball it and say it's exactly 33%, but it's really close to 33%, right? So this is not that unusual a value. And so our summary would be something like this. We're going to not reject. In a study of 38 genotyped individuals, genotypic frequencies did not significantly deviate from the expectations of drawing alleles at random from the single gene pool. And that's the tetraploid form of the Hardy-Weinberg equilibrium is what those genotypic frequencies are. So when I said it was not significantly deviating, that's where I put my chi-square test statistic, the degrees of freedom, and the p-value. In terms of summary stats, this is another situation where we'd probably want to display all this data, these observations and expectations in terms of a group bar chart. So it's just tedious to run through all those numbers for people. So we'd probably have a figure to go along with this, but we don't make too much of these differences because they're not statistically significant differences. Okay, so hopefully that makes sense to folks. We've got a couple of ways in which there are connections between the binomial and the chi-square, but they're pretty distinct ways of using the relationships between these distributions. One, the binomial is just giving us a null hypothesis to test. That was the second, or the values for the null hypothesis to test. That's the second form that I discussed. And that's really common because those kinds of binomial assumptions are really assumptions of no interaction at all, sort of independence of events. So you think there's some process, but the number of counts are just repeated draws from the same process over and over again. no interaction going on. So that's a really common form of null hypothesis. So very frequently the null expectations for some count data comes from something like the binomial, and then we can test it using the chi-square test. And the other thing that we started out the lecture with was sometimes it's more convenient to test, to do a binomial test by using a chi-square approximation, just to save us some computation. All right, hopefully that makes sense to folks, and if not, ask us questions of us in class or email us or office hours.